Who's ready to try some extra credit? You are, maybe. And welcome. Okay, let's go through it real fast, but pretty thoroughly. Here's the extra credit assignment, and here's an example that I've done, at least partially done. So what you're going to do is, this is going to build on our homeworks. Remember the homeworks where you learn something about the 30 families of viruses that are most likely to cause some kind of infection or be associated with humans. Or 31, however many it was. Yeah, that number. All the ones that are in our homeworks are fair game, and you're welcome to update the names. That's fine. The 30 virus families, so you're going to pick one. I'm going to pick coronaviruses because, of course, I'm going to pick coronaviruses. Come on, yeah. Once you've chosen a family, and everyone can do this, everyone can do all of these. There are 4 points available per family, and if there are 30 families or 31 families, that's 120 or 124 points that are on the table. You can do 1 point's worth, you can do 124 points worth, and either way it's fine. We'll grade it, add it to your thing, and it's going to be good. So this is due on the same day as we take the second exam, exam 2. So check your syllabus. All right, shall we? Let's go. So pick a coronavirus, in this case, is what I'm going to do, but you're going to pick one from each family, and that's fine. You're going to write down the name of the virus, and you can write it down in the form that it appears in the paper, or you can go and look up the genus and species if that makes you happy. Yeah. So I'm going to use the form that it appears in the paper, which is this one. Here's how I found it. I Googled it on Google Scholar. That's a nice one, and sometimes you have to put completion of the genome, because for the old papers, they tend to have several where they'll sequence little tiny bits before they actually sequence the whole thing. You need it to be the whole thing. Yeah. All right, and so we found it, and there it is, 1987 MEG Borsnell. Yeah. So we can go to that paper. Here it is. Beautiful. And once you have the paper, what you're going to do is you're going to find first the reference for it. Now, for the reference, I like Google Scholar because you just hit the cite button, double click that, paste it in there, and that's what that is. For the reference, you are going to find the figure, and it's not that one, it's the figure where they actually describe the genome organization. And in this one, they're just completing it, so they don't have the whole genome, but they have the first 2 genes, which is like 20 out of 30,000 bases for the genome, so pretty close. And so I've copied that, and voila. And you can screen capture it, copy it, doesn't really matter, and it's going to be from this paper, so you don't need to cite anything else. You just need the picture, and you need the reference, and I need to know what virus it is. 1 point in the bank already. Did that take you a full minute? Probably not. Yeah, free points. Oh my gosh, like free money. Then for 3 points, this is a little more involved. It's a baby bioinformatics assignment. You're going to tell me a little bit about what sorts of genes this particular virus has. Some of these viruses are enormous, so we're not going to go nuts. We're just going to look at the 5 largest genes that you can find. So the procedure is going to be this. First, you're going to locate the genome sequence. So what I did there was I put infectious bronchitis virus, and I put 2 of the names that were on the paper where they completed the sequence. Yeah, Borsnell, and I put Phil Green on there because he's a good guy. Yeah. So there you go. Or Bins and Green. Yeah, fine. Whatever. These are all legendary people. Yeah. And that takes me to a couple of choices. Now I'm looking at these, and generally it's going to be the one with the long size. You can also Google what's the expected size of this genome if you're not sure which of these to use. But usually it will say complete genome in the name. And so fine, so we can click that. That'll take you to this page. Sweet, and it's going to look like this. All right. From this page, you get your next piece of information. So we've got to put down the accession number. And you can find the accession number right here. Boop. Yeah. And yours will probably be an NCBI reference sequence also, just because there's one of those for pretty much every strain. Unless it's really recent, in which case it may not be a reference strain yet. But someday. Someday we all can. So we copy that down, paste it in here. Awesome. Then we're going to take the genome, and the genome is down here. So this is a NCBI nucleotide page. You can go to ncbi.nlm.nih.gov, and you can select nucleotide, and then you can do a search there, and that will get you to the right place. Or you can Google NCBI nucleotide, and that'll take you to the right page, and then you can just put it in the search bar. Yeah, what are you looking for? Here it is. There you go, click it, and that takes you here. All right, once you have that, you're going to turn this into plain text, and it's called FASTA format, and there's a little button for that. Can you see it? It's right there. Boom, FASTA. All right, it's fast, align, format this bad boy, and so there it is. Now, this is in a format that we can actually use. So we're going to do a little bit of bioinformatics, not much. It's not going to hurt. It'll hurt a little bit, but not much. Scroll. We're going to copy this whole thing. You're not going to need the title line for this, so you can leave off the title line. It's the first line with a greater than at the start of it. Copy. Amazing. And then we're going to go to a program called ORF Finder, which you can find at this address right here. And in the ORF Finder box, you're going to paste your thing. The only thing you're going to change is it comes up with these little tiny open reading frames, and they just annoy me. So we're going to go down here to 300 nucleotide length, which will give you proteins of 100 amino acids or bigger. You'll have some. You'll have at least one. You'll have maybe 100 in some of these big DNA viruses, but don't fret, we're only working with the 5 biggest. And you can just eyeball it. Yeah, get a look at the 5 biggest, or they have a handy little table. Don't change anything else. Hit go. There we go. Wait about a second. There you have it. Now we're going to copy this and we're going to screen cap it. Now I'm using my screen capture program to record this video, so I can't show you how I do it, but I do a find for screen capture or whatever. Your results may vary. You're just going to copy this thing, and voila, I have pasted it here. Amazing. Yeah. All right. Now we're going to take this into a program called HHPred. And there's our result. There we go. HHPred. Let's get you a brand new HHPred page so we can look at that. It's got a nice big blank box here. That's great. And it's going to probably default to this one, which is okay. You're going to paste the protein. How are you going to find the protein? Easy. So on ORF Finder, you can click the large proteins. You can click them down here. They can actually, they'll actually put them in order. So that's the largest one. There's the second largest one. Third, fourth, fifth, and we're only doing 5. Yeah. It also gives you a name for them. Open reading frame 15243. These don't mean anything, but we're going to use them to keep track of the genes. This is not what anybody else calls it. This is just what ORF Finder is slapping onto it, but it gives us a point of reference, so we're going to use it. So you're going to have the little picture here, and it's going to say, if you zoom in, look, ORF5, wow, is this one. So this is polyprotein 1A, 1B, that's the spike, that's the M, that's the N. This is the E, but we're only doing the 5 biggest, and E is not among the 5 biggest, so too bad. E, you don't get to play. All right. HHPred, you're going to, one at a time, copy these proteins. So let's get the first protein. There we go. And this is the sequence of that protein. You don't need the title. You just need the sequence. So the sequence is going to be amino acids. It's going to look like all the letters except 6, I think. There's no B and there's no J amino acid and there's no O and whatever. Yeah, you get how amino acid sequences work. We're going to copy that. We're going to take it over to HHPred. We're going to dump it in the box. Then we're going to add 2 databases. So it's going to work best if we add the PFAM-A database. We'll keep the PDB default. That's at the top of the list if you delete it for some reason. And we're also going to get NCBI conserved domains. Now, they update this database every couple of weeks. And so yours may be a different version number than the one you're seeing in this video. That's okay. Yeah, that's progress. That's expected. That's actually good. It means you're working with the most up-to-date version. But there's going to be a PDB, there's going to be a PFAM-A, and there's going to be an NCBI conserved, and you're going to locate those. Put them all in here. That's great. And now you're going to hit submit, just like that. I already did it. At fast times, this is going to take maybe a minute, 2 minutes. At slow times, this may take you 20 minutes. You can run several of them at the same time, though, but I wouldn't run more than maybe 3 at once, just because there's a memory leak thing with some browsers, and it'll make your browser very sad and your computer very slow. But you can always close out of it, Yeah, goodbye, HHPred. And you'll have a list of your jobs, recent jobs on the side. And so my last job is this one, and there it is. So this is opening reading frame 1A from infectious bronchitis virus, the coronavirus. Now you're going to interpret this. You're going to look for columns. So this is like the start of your sequence. This is like the end of your sequence. It'll highlight some region in the middle that it feels like doing. Whatever, you don't have to, So the entire set of amino acids that I put in, just from reading from 1A, was 3951 amino acids. Good for me. We look for columns, so I see one column here, 2, 3, 4, 5, 6, 7, 8, column 9, 10, 11, 12, and 13. And you notice when I mouse over these columns, it's going to tell you a little bit about them, and that's fine. So once you've got that, and it'll have a message here at the bottom, you click that message, the magic chevron will appear. The magic chevron will always take you to the top of the page, because we're going to be way down the page for some of these things. So bam, we're going to go. First one, which parts did we align? Well, you can tell. You're going to look at the second line. It's going to start with the letter Q for query. This is the question you asked, the protein you pasted in. Which part of it did it find? Start over on the left side, work your way to the right, so you'll be starting closer to the beginning of your protein. We're going to start at amino acid 1. Awesome. You're going to make me a table. You're going to say, okay, open reading frame 1 from or finder. And we're going to say the first domain or part of this protein, the first column of things that it can be, is going to start at 1 and it's going to end at, and now you see how this is hit number 2. We're going to go down till we see hit number 3 appear. There it is. And then we're going to go over to the end of line 2 here. This is just the end of that protein. It's broken it into chunks like that. So it's still going to be the second one right there, and look at that. That's where we end, 673. And it says the total is 3951. We already knew that. We don't need that information. So 1 to 673 is a match for non-structural protein 2 from IBV. And so that's going to be our annotation, IBV non-structural protein 2. And the actual thing we're matching is going to be this one. It's this little green link. That'll take you to the protein structure or protein family or NCBI conserved domain, depending what you hit. So 7F52, and you can have underscore A, or you can leave off the underscore A. It's fine either way. Let's try the next one. Now, when you're confronted, so to go to the top, hit the chevron. All right. When you're confronted with something messy like this, it's okay. Use your judgment. I found some big domains that cover it and some little domains that are just parts of it. You could do either. Generally, I'd rather diagram this in smaller parts, but yeah, I think for this one it's fine. So let's go with just this column because I like the look of this. I'm going to avoid this big long one because I want to look at each of these columns because each one of these does its own job. They're each different enzymes or parts, and so I would like to see those separately. All right, so now here we go. Let's get this one. Great. Non-structural protein 3, and it says it's either an X domain or a macro domain. You can see that down on the third line. It usually takes the description a little while to get going. So great, X domain or macro domain, and I've already written it in the table here. You can use my table, and actually if you're doing a coronavirus, you can just wipe out the first 3 columns and use the 4th column because it's going to be the same. Yeah, and it's going to be fine. Let's go down there and look at that. 3EJF. There we are, 3EJF. You remember the drill. Where do we start? 1005. All right. So 1005. Where do we end? I don't know. Let's go see. So we're at 119. So let's scroll down. There's 120. Go to the end of the second line. There we are. 1176. So this means that in this protein, which is 3,500 amino acids long, whatever that number was, or 3,900, the first 673 are going to be NSP2. the job of the 1000 to 1176th amino acids, somewhere right in here, is going to be that's going to be the X domain. And so you can get a feel for where all these things are and what sort of equipment each virus is bringing to the party. Yeah. All right, because that's virology. That's how you understand these viruses, actually. What did we match? I don't know. Let's go see. What did we match? We matched 3EJF, great, underscore A. So that's a protein structure. 3EJF, A, awesome. And that's its title, and so on. We're not doing the first 5 domains. We're doing the first 5 proteins, and we're doing all the domains in the first 5 proteins. Yeah. All right. Anyway, complete the second section, and you get 3 points for each virus family for which you complete this. So with that in the first section, potentially you got 4 points per virus family times around 30 families, or whatever you want to call them, like hepatitis delta virus. That's not a family, but it's in a family, but whatever. Yeah, and so go rack up the points. If you want to work with somebody, it's fine, let me know, and because Turnitin will pick up that 2 of these are exactly the same. Yeah. And I can split the points between you if you want to do that. But why not have all the points for yourself? Like, work together at the same table, help each other out. Like, oh, how do I do this? How do I find that? Yeah, sure. Pick a different virus. It's fine. It's easy. It's no big deal. Yeah. So there you go. You can do this. This is the extra credit. And I'll give you this assignment sheet with the example embedded in it, because I think that's a classy move. And there you go. Enjoy. Thanks. Bye.